Welcome to State of the Science. And the topic today, this is the first of 6 series. And the topic today is T1D or type 1 diabetes one disease or endotypal? What's the variability of the T1D phenotype and what's the evidence? So I'm going to do an introduction for our distinguished panelists and talk a little bit about what this meeting is. So the State of the Science is really just a new way of talking about research in the field of type 1 diabetes. This series is going to be a celebration of the power of debate and discussion that women scientists in type 1 diabetes research bring to the table. We have a global audience of scientists here today, and we thank everyone for joining. I'm going to briefly introduce the panelists. These panelists will set the stage for the discussion, and then we'll ask them to weigh in on their thoughts. And on, we have 2 sort of polls that we're going to engage with the audience, and conversation is going to be free flowing, somewhat like a cross idea exchange, a town hall, and a respectful debate. We're going to pause at 2 different time points to conduct polls with the audience and have the panel weigh in on the audience voting feedback. So there's a couple of pro tips. If you want to submit a question, put it into the Q&A. You can submit questions throughout the presentations, and participants can upvote your favorite questions. At the end of the session, we'll answer questions as time allows. The event's being recorded, and we will post the recording later on our Sugar Science YouTube channel. So in the interest of saving most of our time for discussion, I'm going to do very brief introductions for each of our scientists. We could actually take up the whole time discussing each of them. They're super valued and integral members of the type 1 diabetes research field, and you can connect with them directly after the presentation. But I'm going to start with Dr. Linda DiMeglio, and she has she may join at the very end, but she's been called away. She works in clinical research with a focus on type 1 diabetes prevention, beta cell preservation, and new technologies and therapeutics. Dr. Carmella Evans-Molina, she's the J.O. Ritchie Professor of Medicine. She serves as the Director of Indiana Diabetes Research Center and the Director of the IDRC Islet and Physiology Core. She directs the Diabetes Research Program in the Herman B. Wells Center for Pediatric Research, and is a staff physician. I don't know how she's doing all this stuff. She serves as an associate director of the medical scientist training program at IU, and she's an investigator in type 1 diabetes trial network, where she serves as scientific advisor and as chair of the long-term investigative follow-up LIFT study. I could really go on and on here, but suffice it to say, she has deep roots in this community, in the research community, and she's a leader in the field. Dr. Bridget Wagner is the Director of Pancreatic Cell Biology and Metabolic Disease in the Chemical Biology and Therapeutic Sciences program at the Broad Institute of MIT and Harvard, where she's also an institute scientist. Her group's research focuses on the chemical biology of diabetes with the aim of identifying small molecules capable of increasing pancreatic beta cell number and function, and the ultimate goal of discovering new therapeutic approaches for diabetes. Dr. Emily K. Sims is a pediatric endocrinologist at the Riley Hospital for Children and a physician scientist at Indiana University Center for Diabetes and Metabolic Diseases. Her research passion lies in identifying and understanding risk factors predisposing susceptible individuals to diabetes development. Dr. Sarah Richardson, PhD, she's coming to us from England, where she is an associate professor in cellular biomedicine at the University of Exeter Medical School. She has a passionate interest in type 1 diabetes. She's been awarded 3 prestigious fellowships and more recently the JDRF 5-year Career Development Award. And she was the very first recipient of the NPOD Junior Investigator Award. And she also co-leads the Islet Biology IBEX group. Her research utilizes 3 unique cohorts of type 1 diabetes patient pancreas samples and is centered around developing a clearer understanding of the disease process. Dr. Georgia Fousteri. Dr. Fousteri studies the mechanism that control autoimmunity. She investigates, her group investigates the genetic and cellular factors that set the immune system in equilibrium. And they're also developing innovative approaches to promote tolerance in patients with type 1 diabetes. She's also coming to us from Europe. Dr. Lucy Walker. Dr. Walker is a chair of immune regulation at University College London. Her main areas of expertise are T-cell differentiation and regulation, T-cell B-cell collaboration, autoimmunity, and type 1 diabetes. And her group has spent many years studying how regulatory T-cells inhibit autoimmunity, focusing in particular on the mechanism of action of the immune regulatory protein CTLA4. And finally, and not last but not least, Dr. Jessica Dunne. She was formerly at JDRF in a leadership position, and now she's currently directing the screening initiatives at Johnson & Johnson. She was also one of the initiators and authors of a very important new paper, The Women's Leadership Gap in Diabetes, a call for equity and excellence, that was published in Diabetes Care in 2021. Distinguished panelists, thank you so much for joining us. And would you set the stage for this discussion? Is type 1 diabetes one disease or endotypal? I'd ask Dr. Fousteri to share her slide.
Okay. Can you hear me?
Perfectly.
Okay, perfectly. So before, I guess, responding to this question, I'd like first to thank you for the invitation and all the panelists today, Women Power, and everyone in the audience. Hopefully we'll be able to answer some of your questions and perplexities about this theme that we're discussing today. So we thought that to initiate the discussion by actually clarifying and what is an endotype, and define the difference between a phenotype and endotype, particularly for type 1 diabetes. So phenotype are the observable characteristics of the disease that does not imply or implicate mechanism. For example, type 1 diabetes are phenotypic characteristics as the fact that the patients are insulin deficient, they have glycosuria, they have diminished C-peptide levels, they increase HbA1c levels, and often islet antibodies. So endotype, what it means is a subtype of a disease condition that is defined by a distinct pathophysiological mechanism. For example, it's the mechanism that is underlying the disease, and there we can have different endotypes. And with regards to type 1 diabetes, and many of you have heard of the islet reactive T cells, or whether or not it's a Th1 driven disease, there is a role of neutrophils, Tregs, follicular helper T cells, cytokines, B cells, autoantibodies, etc. And we'll also hear today by the other panelists and how identification and studies on this type of cells have led to the definition of 2 endotypes so far for type 1 diabetes. So actually endotype is what is the mechanism that actually underline the phenotype of type 1 diabetes. And this mechanism and these cells or soluble factors, let's say, are called biomarkers. And there's an increased need in developing biomarkers that are easily to measure, particularly in the case of type 1 diabetes in the peripheral blood. And because, it could actually help us through this analysis predict the response to the treatment and therefore allocate patients to clinical trials more precisely. This is what is called, precision medicine. But also these biomarkers could also help us forecast disease risk and progression, and therefore be able to develop strategies that would be efficient at preventing or delaying the disease. And with this introduction, I ask my co-panelist Sarah Richardson to share her exciting data on the endotypic analysis of type 1 diabetes.
Thank you so much. That was
Thank you,
excellent.
I've just organized sharing my screen. And pop that in. Can everyone see that okay?
Perfect.
Yep. Okay, so thanks, Georgia, and thank you, Monica, for the invitation to this really exciting panel. I'm really looking forward to discussing this with everyone today. So as you've already heard, we're really aware of the heterogeneity in type 1 diabetes, in that we see differences in the progression from autoantibody positive to type 1 diabetes or loss of C-peptide after diagnosis, and in responses to treatments. And so what I want to share with you today is how our pancreas pathology studies that have been made possible through our access to unique Exeter archival diabetes biobank and other contemporary pancreas collections like NPOD and DIVID, and how these have revealed 4 key age-dependent differences in type 1 diabetes pancreas pathology. and then how these findings may suggest that there could be different underlying disease mechanisms despite the single outcome of a requirement for insulin. So these age-dependent differences that we've observed relate to the types of immune cells present, the absolute numbers of immune cells present, the levels of beta cell destruction, and the processing of insulin. And for this all started with an observation by Pia Leitz during her PhD, where she noted that there was a subset of donors who have evidence of many CD20 positive B cells in and around their residual insulin-containing islets, or ICIs. However, in a second subset of donors, these CD20 positive B cells were vanishingly rare. Some examples of this are shown here, where the CD20 positive B cells are identified by the brown stain and the CD8 positive T cells by the pink stain, and the islet is shown in the dark blue. And based on these observations, we split our cohort based on more or less than 3 B cells per ICI. And when we did that, these age-dependent differences became really apparent, particularly when we plotted the average number of CD20 positive B cells per insulin-containing islet against the age of onset of type 1 diabetes in nearly 70 type 1 donors with a duration of less than 2 years, most of them with a duration of less than 3 months. And these samples represent some of the rarest material available in the field. So if we look in the donors that were diagnosed under 7 years, 15 out of those 16 had more than 3 B cells per ICI. But in contrast, if we looked in those donors diagnosed over 13 years, 29 of the 30 had fewer than 3 B cells per ICI, whereas those diagnosed between 7 and 12 were split between the two. And really, it was what Pia discovered next, though, that really helped us to cement the idea that we might be dealing with 2 different forms of diabetes. So whilst examining the localization of the pro-hormone form of insulin, or proinsulin, shown in red here, and the mature form of insulin, shown in green, Pia found that in some donors, proinsulin and insulin were located in 2 distinct subcellular compartments. And that's the staining distribution we typically see in beta cells with individuals without diabetes. However, in others, she observed far more yellow staining, indicative of a co-localization of proinsulin and insulin in the same subcellular compartment. And this suggests that in these individuals, the insulin is being abnormally processed. Importantly, we were able to show that all those donors with high numbers of B cells also had this evidence of abnormal insulin processing. And those with the more typical distribution of proinsulin and insulin had fewer than 3 B cells on average in their ICIs. And this led us to propose that there are 2 diabetes endotypes, or forms of disease. Type 1 diabetes endotype 1, or TIDE1, where donors have more than 3 B cells per ICI and evidence of abnormal insulin processing and are diagnosed young. And type 1 diabetes endotype 2, or TIDE2, that have fewer than 3 B cells per ICI and a more normal processing of insulin, who tended to be diagnosed older. When we split the donor cohort based on these endotypes, this helped to reveal further differences. And so here we have 2 representative pancreas sections from TIDE1 donors in yellow and TIDE2 in blue. And the islets in the sections have been annotated to identify those that still contain beta cells that are shown in red or green, and those with no remaining beta cells or insulin deficient islets in purple and blue. And those with evidence of immune cell infiltration, defined as 15 CD45 positive cells, are shown in either green for the ICIs or blue for the IDIs. And what I hope you can see is twofold. Firstly, the number of green islets, so the ones with beta cells and evidence of infiltration, are increased in the TIDE1 donor compared to the TIDE2 donor. And secondly, that the number of red islets, the ones that contain beta cells which have limited evidence of infiltration, are increased in the TIDE2 donors. Now, this can be seen more clearly in these sunburst plots, where the inner circle identifies the proportion of insulin containing islets, and the outer circle demonstrates the proportion of the islets that are inflamed. or not in each of the different categories. And so we've now been able to analyse more than 10,000 islets across a large number of these T1 and T2 donors. And this has revealed that T1 donors, shortly after diagnosis, have significantly fewer islets that still contain beta cells when compared to the T2 donors. And across over the 5,000 islets assessed in each of the endotypes, we find only 10% of islets that still contain insulin in T1 donors, compared with 31% in T2 donors. And in separate studies combining data from NPOD and EADB, completed by Alice Carr, a PhD student in our lab, we found that 88% of those diagnosed over the age of 13, so T2 donors, still have some residual beta cells up to 5 years post-diagnosis. And this compares with only 8.3% of donors diagnosed under 7, or the T1. So this really suggests that that increased inflammation that we're observing in T1 is resulting in a more rapid destruction of the beta cells. So we feel our data suggests that although type 1 diabetes has one endpoint in that individuals diagnosed will require insulin, the pancreas pathology hints that there are two different endotypes or forms of diabetes, and we've termed these T1 and T2. T1 is characterized by younger onset, especially under the age of 7, with more immune cells in more islets, and this includes high numbers of those CD8 positive T cells and those CD20 positive B cells. They have fewer residual beta cells at diagnosis, which are lost rapidly, and evidence of abnormal insulin processing. In contrast, the T2 donors are older at onset, typically over 13. There are lower numbers of infiltrating immune cells, very few B cells, and beta cells are retained for long periods. Key, though, is the observation that donors between the ages of 7 and 12 fell into one or other of these pathological endotypes, lending more support for different forms of the disease rather than a continuum of change. So we feel that this gives important insights into type 1 diabetes heterogeneity that are emerging from the study of pancreata from these rare short duration donors. And we think this has important implications for both the monitoring the progression of the condition and for more tailored and effective treatment choices in the future.
That's really excellent. We had one question, whether or not the sections are imaged from the same region of the pancreas.
So, sorry, could you repeat that?
Sure. I'm just wondering from the audience, from Stacy Weaver, are those sections that you showed, or were they imaged from the same region of the pancreas? The two sides.
Yeah, thank you. So unfortunately, with the EADB biobank, we are not entirely sure where these samples have come from within the pancreas most of the time, but they are the only samples that are this close to diagnosis in the world. So we have to use what we have.
Thank you so much. And now can we hear a little bit from Dr. Walker?
Great, thank you very much, Monica. Let me just share my screen. Okay, how's that?
Perfect.
Great. So thank you, Monica, for the opportunity to be part of this discussion today. So I just put together a couple of slides to highlight my thinking on this area, which is basically because type 1 diabetes is complex and polygenic, like all autoimmune diseases, because there's a role for environmental influences like infection, for example, I think inevitably we are going to see heterogeneity. So we're going to see people that have ended up in the same endpoint, so insulin-dependent diabetes, but for different mechanistic reasons. And if we think about the sort of parameters we might want to consider when subdividing people with type 1 into different categories, obviously we have genetics, we have antibodies to beta cell antigens, and we can look at the number, the specificity, and the order in which these appear. There's very exciting data around the age of onset that Sarah has just talked to us about, this idea that individuals developing diabetes under the age of 7 have this different qualitative phenotype, more CD20 high, pancreatic infiltration, more aggressive destruction of the beta cells. This boxed area is where my focus mainly lies, which is understanding the loss of immune tolerance that leads to the development of diabetes. And we may be able to divide people into different camps based on their immune phenotypes in these sort of areas. So we can look at immune regulation by looking at regulatory T cells, these T cells that suppress immune responses. We can look at number and function of these. We can look at production of and sensitivity to interleukin-2, an important regulatory cytokine. We can also look at the effector T cells. We can track autoantigen specific T cells, and we can look at T cell differentiation states. And we've done a little bit of this work looking, for example, at follicular helper T cells, which I'll just talk about in a minute. So I think broadly we can stratify based on mechanistic understanding of disease processes, and we also have another tool, which is to look at response to therapeutic intervention. So we can divide people into responders and non-responders when we treat with particular therapies. So just an example of the mechanistic understanding. So we can consider CTLA-4 haploinsufficiency, which is something that we reported a number of years ago now. So we described individuals who had mutations in CTLA-4, meaning that they had less of this regulatory protein in their Treg, their FOXP3 positive regulatory T cell population. And these individuals can develop lots of different types of autoimmune disease, and some of them develop type 1 diabetes. And you can envisage that if you develop diabetes because of a problem with your CTLA-4 pathway, then you're likely to be responsive to treatments that act on that pathway. So putting back CTLA-4 function, for example. In terms of considering response to treatment, I just wanted to give one quick example of some of the work that we've done looking at follicular helper T cells, or TFH. So these are T cells that are specialized at interacting with B cells. They support B cell antibody production. And we found a few years ago that individuals with type 1 diabetes, if we look in their memory CD4 T cell compartment in peripheral blood, we see a signature for follicular helper T cell development. And because these T cells are quite sensitive to CD28 co-stimulation, we looked at the follicular helper T cell population in individuals treated with co-stimulation blockade drugs, so abatacept. And you can see that people given this co-stimulation blockade drug had a reduction in follicular helper T cells at year 1 and at year 2 compared to individuals who received placebo who didn't show a change in TFH. But most interestingly, when we looked at the TFH profiles in people before they were treated, we saw that there were differences in the individuals who went on to be clinical responders compared to those who went on to be non-responders. And I'm not going to go through the data in any detail, but you can see here that people who showed a poor response to immunotherapy had a higher percentage of ICOS positive TFH. Now this needs to be validated in another cohort, but if we take this at face value, if this turns out to be true, the question is, does this reflect a fundamentally different disease process going on in these individuals compared to these individuals. And theoretically, you can come up with possible plausible biological mechanisms for this. So we know that ICOS positive TFH produce more of certain cytokines than ICOS negative TFH. Could it be that these cytokines are substituting for CD28 co-stimulation in these individuals? So I guess by studying responders and non-responders after therapy, it may be that this can reveal, in some cases, distinct endotypes that are underpinned by different mechanistic disease processes. And of course, this feeds back in both directions, because a better mechanistic understanding of the disease process that's applicable will inform the best biomarkers to use and will help predict response to therapy. So I think there is heterogeneity. I think it's complex. It's going to require a combination of different approaches to unpick this, so immune phenotypes in combination with a number of the other parameters that I showed initially. But I think this is worth doing because of the potential to impact treatment strategies. So I will leave it there. Thank you, Monica.
Fantastic. We have just a quick question from Annette Ziegler. Very nice. She says, and do you know whether those responders versus non-responders are also genetically different?
So we don't have genetic information on those individuals. Yeah, I think that would be very interesting to add to our data set. Great.
There's a longer question here. I just wanted to check to see that the panelists can see this question from Matthias van Hereth, and if one of the panel would like to respond to it. Basically, he's saying that the view on endotypes and further subclassification of type 1 diabetes from the pharma perspective is a very cautious one. From what I see is that especially in younger patients, the disease develops faster, but age, of course, anyway, is of course anyways a factor in trials such as BMI and gender. However, as the business case for T1D prevention is not ample, I would at this stage be very careful not to slice a small pie into ever smaller pieces. It's not helpful, especially if companion diagnostics would be required. So that's sort of like, the other side of the coin, I guess. How might the panel respond to that?
Maybe I'll take the first stab as also being on the same side of this, and maybe the only one of the panel that's on the other side of this endotype question. And I think about the name of the panel is T1D one disease or endotypal, and my answer to that would be no. I think it's something else, right? So I think Matthias brings up some really good points in terms of, not to discount the beautiful data that both Sarah and Lucy showed, but I think also what they did in their talks was also point to, and Lucy made a very nice point about this, the prospective validation of some of the work that's been done. How do we make treatment decisions on this? So when I look at the panel, how do we make individual decision points on this? When we think about the fact that maybe this year we will have one disease modifying therapy on the market for type 1 diabetes, and how long did it take us to get there? So if realistically we're starting with the next therapy for T1D, it's going to take us right now to look at the entire people of, let's say, in stage 2 T1D, 10 to 15 years to get to the next approved therapy. So now if we're starting to slice and dice the population, we're looking at longer timelines. Now, what I'm not saying in all of that is, do we stop progressing the science? No. So what I'm trying to suggest is that maybe we take a little bit, and I think this is what Matthias is suggesting, is we take a little bit more practical approach, and instead of saying, and I think this is really what I'm reading from both Sarah and Lucy's data, is that maybe there's something here about we've got to validate these in larger cohorts. We have to be able to make decisions at an individual basis, but at the same time, we can't get so driven down deep into this idea that there may be ways to intervene in these small subpopulations by forgetting the bigger picture. So that's why I say, is T1D endotypal or is it one disease? And my answer is no, because I think we've got to look at it from all sides here, right? So if our hope is to really drive therapies to the most people as quickly as possible, then I think we've got to start from a basis of starting at that point of looking at the entire disease. And then I think, as Lucy said at the end of hers, you know, we start really driving down mechanisms in clinical trials to start really understanding the mechanistic basis of what is driving T1D. So we're probably all saying the same things in different ways. So
yeah,
maybe
no,
just to add some more color to Matthias's point.
No, I think it's great. You're coming from industry. And so that's what you guys are thinking about, getting something of value to market to patients. But on the other hand, in academia, it's really a quest for understanding the etiology. And I mean, there is this whole idea of personalized medicine. So how can is there room in the conversation for that? You know, like where does that come in? I mean, medicine doesn't seem to really be there yet. I don't know. I mean, you guys have to answer that, not me.
Yeah, can I make a comment about that too, Monica? I'm a fan of maybe not endotypes, but phenotypes, or like thinking about heterogeneity in general, but I don't know. Okay, so another big question that kind of is to like Matthias's point is like this relationship with age. I think we don't totally understand, like almost every kind of subgroup that people have identified as linked to age, like younger age somehow. And I don't think we totally understand this idea of like the continuum versus bends, and whether, you know, some of these are just reflecting like more severe disease that presents in little kids, right? So I think that's one thing that we really need to understand better. And I agree with this idea of kind of moving that science forward to understand like how these are really linked to pathology. But then the other thing that I was going to say is, I'm not sure that, you know, this point about the kind of pragmatically like not cutting people off from treatment so that we can continue to move therapies forward has to exist separately from the idea of continuing to explore like the possibility that there are subgroups that we can treat a little bit differently. Or the other thing is, it may mean that, you know, we have to think about the timing of when we intervene in these different populations differently, right? So it may be that we have to think about it that way too, versus this is a group of people that's going to respond and this is a group of people that isn't going to respond. So I guess I agree with the point that just all of the science needs to move forward. And I don't know that it necessarily has to be something where we're kind of excluding large groups of individuals. But I mean, I think that the cancer field, at least, you know, there's been lots of examples of how people have identified these subgroups where you can use more targeted approaches, and they've been really effective. And there have been companies that have been willing to kind of develop that too, right?
Yeah, and actually, I think I was actually going to bring up the cancer as a great model, right? So if you think about it, if we never had chemotherapy, where would we be with cancer, right? So that's, I think, the approach that I think about it, right? So yeah, so I think it's a good model, working model. I think we're sort of saying the same things in different ways, Emily, is that, right? So both of these things should advance, and cancer is a great example that if we didn't have we haven't had chemotherapy for the last 20 or 30 years, would we have these more targeted approaches today? It's an interesting philosophical question that I don't know that any of us can answer, right?
Let's take a moment for to poll the audience and get their thoughts. So I'm going to launch this poll. The poll 1 is, how does phenotype variability affect the strategy of developing new treatments for T1D? And I'll launch it and we'll see what people say. So the first answer is PV, or phenotypic variability, is incredibly important variable to be considered in designing new treatments. The second response is, it should be increasingly prioritized in clinical trial design, A and B. And then PV is somewhat important or it's not important. So it looks like we can see in live time, I hope all the panelists can see this, that it looks like A and B are coming in sort of strong, but who knows? Let's see, we'll give it up to 80% if 80% of the people have voted.
I'm kind of disappointed that I don't get to vote on this.
Yeah, me too. I did try pressing the button.
Can you not vote? That's terrible. Oh.
No, it says host and panelists can't vote. I did try pressing the button too.
Oh no, I've locked you guys.
Yeah, I know you did. Yeah.
Well, I mean, you'll get to, after we do this, after we get the results, I'd love to hear what you think sort of one by one, and that will be a good opportunity to to come out and share. Okay, so it looks like, it looks like basically people in the audience, in the scientific audience, majority scientific audience, are saying that it looks like, phenotypic variability is incredibly important variable to be considered in designing new treatments, and it should be increasingly prioritized in clinical trial design. So that's something. So what say you about all that? Anybody? Don't know. Jump.
I was going to vote somewhat, actually. I mean, I agree with the point that we need to be kind of developing broad therapies and just moving into getting treatments, especially for prevention that exists that are accessible to people. But I mean, I definitely agree. This is important science that we need to move forward and be exploring. I think Lucy has her hand raised too.
Oh yes, go for it, Lucy.
Yeah, so I was just going to say in response to Matthias's question that I think there's what we have now, and then there's thinking about what we will want in the future. And I completely agree that in terms of making a business case for now, it's important actually not to slice this small pie even smaller. But if we look forward to a situation where there might be a choice of different immunotherapies available for people with type 1 diabetes, at that point, we're really going to want to understand these mechanisms much better and understand how to select the right immunotherapy for the right patient group. So there's sort of what we have and can do now, and then there's also preparing for the future.
Well said.
I have an additional.
Sorry, go ahead. I was just going to say, I think another really important thing is going to be like reliability of these biomarkers that we use for these different endotypes. A lot of the analyses we've looked at have been cross-sectional, including a lot of the analyses I've been involved in, right? And so I think we're really going to have a better understanding of, in a person who's at risk for type 1 diabetes, like how does this biomarker change from day to day, longitudinally, what's the variability there? Can we just look at one point in time and make a good assessment, so that we can use those, for clinical decision making.
Yeah, that's a good point. I mean, some people can show up with GAD autoantibodies and then never even go on to develop type 1. So perfect. Did someone want to say something?
I think both Sarah and I have our hands raised, but she was first. So
okay, keep an eye on whoever put their hand up first if you can, and then just jump in.
Yeah. Thanks, Jess. Yeah, I was just going to say, I completely agree with what you just said, Emily, about the biomarkers and particularly what we're seeing within the pancreas. We need reliable windows as to what's going on in there, because we can't look at what we are doing in everyday people.
Yeah, and then just I completely agree with what Emily said, and the point I was going to make too was a pragmatic point on just to follow up what Emily said is, from a regulatory perspective, as we start looking at some of these biomarkers, start understanding what that might look like in terms of a regulatory perspective, the sooner that we look at it under that lens and understanding what the regulators might be looking for as we think about, especially if it's going to be needed as a companion diagnostic or as a response to treatment, and making sure that we're collecting our data in a way that enables that regulatory piece. So start having those conversations now and understanding what that might look like will help us, from moving down the line, because the last thing you want is to do all this work and then realize you've got to go out and do another 5 to 10 year prospective study, right? Because you haven't collected your data in a way. So this is my plea, those of you that know me well, to make sure that as you're writing your consents, make sure that you are allowing for things like secondary use of their data for these types of purposes, and everybody should be open to sharing their data to the extent possible. So thank you. My public service announcement for the day.
I like that. Viva open science. Okay, so we have another poll too. I'm going to send out the second one. This is the second of 2. And so this poll is basically, what does the phenotype variability PV tell us about the human genetics of type 1 diabetes? So it's like, well, does PV continue to inform us that personalized medicine should be considered seriously as a way forward? The second answer is PV suggests that T1D is an endotypal disease. And the third is that PV has serious limitations in informing us about the genetics of T1D. This is kind of a very interesting question because, it appears to me sort of, as an outsider, I guess, that sometimes the genetics are not always wedded to the biology. And I guess maybe, you know, I'd love to hear the opinions from the audience and the panelists on this one.
Yeah, I can say something, or at least a little bit about it.
Yes, please.
I'll make it a little bit simple. So we know that approximately 10 to 15% of patients with type 1 diabetes don't have autoantibodies at the time of the disease diagnosis. Okay, and we know from works on genetic testing in these individuals that in HLA, for example, that people, most of the people in the audience understand, they don't have the type 1D HLA risk. And some studies that have been conducted by Exeter has shown that these individuals, a significant fraction of them, approximately 30 to up to 50% of those patients, have mutations, pathogenic mutations in MODY genes, suggesting that this type of diabetes is not the autoimmune classical polygenic form, but rather a monogenic form of diabetes. So this, I think, it's very important. So here we have a phenotype variable, which is a number of antibodies, and that actually tells us that we're talking about a different form of the disease. Call it endotype of the domain. And we also have undertaken some studies with regards to this aspect in our clinical center here, and performed clinical exome sequencing in 63 individuals that at the time of the disease diagnosis, 48 of them did not have autoantibodies, and 45 of them, and 18 of them had just a single autoantibody present. Okay, so by undertaking this kind of analysis, you know, exome sequencing, etc., we confirmed the data from Fousteri and the group from Exeter that the autoantibody negative, approximately 30% of them have MODY mutations. But we also found mutations, MODY mutations, and variants of unknown significance, particularly in the ones that have a single autoantibody present, suggesting that, you know, we should take into consideration some phenotype variables when we diagnose the disease. And perhaps this should be taken into consideration when we are planning on treating patients. So I think genetics should be, I think it's a necessary step, in my opinion, before including patients in clinical trials.
That's well said. And here's the results of the poll. I'm going to share them to you guys. It looks at, you know, kind of the first answer is the one most people chose, that phenotype variability continues to inform us that personalized medicine should be considered seriously as a way forward. And that kind of goes to what you were saying. Is anyone else would like to, you know, weigh in on that? Yes, no, crickets.
Well, I have a question maybe for Sarah. I think that it's sort of following on what Georgia was suggesting from that really interesting data that you summarized. You know, as a beta cell biologist, you kind of wonder what role the sort of fragile beta cell plays in some people's disease development. And I was really intrigued about the association between inflammation and the dysfunctional proinsulin processing that you've identified in the tissue section, Sarah. So maybe kind of following that a little bit, do you think that is just a marker of the more robust inflammation, or do you, which is the chicken, which is the egg there, if you could speculate?
That's a really great question, and something we're kind of trying to drill down on a little bit more. I suspect it might be the chicken, the sort of abnormal pro-insulin insulin processing that's going on there, and that's driving the recruitment of the immune cells. But you know, it's a great question and needs to be answered, but it's hard to find the samples in which to try and address that.
Great. And Emily was, because she loves to talk about proinsulin, so I want to give her a chance to respond to that, because she had a very excited look on her face.
Yeah, no, I was just thinking maybe Sarah can like write like a commentary on my next like grant revision in the introduction section about how important proinsulin is. I don't know, maybe I'll talk to you offline.
Good. Sparking collaborations left and right. Excellent. Or anyone in the audience, who has that kind of interest, maybe reach out to each other. Yeah, so at this stage, I think that, maybe it might be nice to open it up to questions from the chat and some in-person questions. And if there's no questions, hopefully there'll be a few, but if there aren't, what is the future, like the go forward path look like in terms of using these laboratory data to streamline the the way forward to get medications to the clinic? It seems, I don't know, maybe this is good for Jessica to answer, because I wonder if it seems like there might be some disconnect between laboratory and industry. And how do you have any ideas for how to streamline that or connect it better?
I don't know that there's necessarily a disconnect. I think the pharmas that are operating in this space all have close ties to the academic community, and I think that there's a lot that we're leaning on you guys to do a lot of this early stage research. So companies like J&J are not developing their immunological targets for T1D, right? So they're developing them for other indications and hoping that the science that you guys are all doing leads us to realize what we can be doing out there. I think, this idea, and one example of such is this work that we've been doing with the Critical Path Institute on the qualification of autoantibodies. So these ideas around public-private partnerships to advance the science, this idea of open dialogue about what the needs are of both groups to really drive the science forward. I think investment in early stage discovery research in T1D. So a plea for our funders to not always fund the fancy shiny ball that you see at the end, but also think about some of this really basic critical understanding of disease mechanism science that I think if it today showed anything, it's that we still don't have a good understanding of how T1D develops. And so we need to unravel that. I think just closer collaboration, and I think we're doing a good job. Like I said, I think all the companies really actively developing therapies have close ties to the community, and keeping those lines of communication open and realizing that we're scientists just like you too, and we're not the evil empire. So, and data sharing as much as possible.
Definitely not the evil empire, because you're getting, you're interfacing with getting these, the medications to the population. I guess I just was talking about okay, we don't want to slice up the pie, or I guess in terms of Matthias's business case for T1D prevention, how do you connect a business case, I guess, to what's happening in the actual laboratories? And you made a good case for that's actually happening, and I love this whole idea that what CPATH is doing, the public-private science, very important and very good. But I don't know, is there room for more conversation there, like that people can talk about it and really get to a point like, okay, this is what industry needs. How can we get there so that we can accelerate the work or accelerate the drug development?
Yeah, and I think that there are maybe two separate questions in there. And one is the business case, which is the business case, right? And that's people way above me who make those decisions on whether or not the business case is worth investing in. I think what you probably hear underlying what both Matthias and I are saying is that, T1D is a relatively small market, and we can't charge for a disease modifying therapy what a cancer immunotherapy can charge, right? So that's comparing apples and oranges. And so understanding what that business case might mean, and now if we say, okay, we're not going to treat making up a number, 500,000 people a year with our disease modifying therapy, but now we're gonna start treating 20,000 patients a year, but we can't charge a different price because that's all the market's going to bear. I think that's kind of the point that Matthias and I are making. Then the business case goes away.
Yeah.
Now, I think the point that Lucy raised, that once there, I think it was Lucy earlier, that once there are multiple therapies on the market, this becomes, and this is where this usually happens, is sort of in this post-market access, is that when then you start understanding who, the right patient at the right time. And that's when that sort of starts. And so I think, again, I think this idea of collaboration, and those of you that have worked with me over the past 18 months that I've been at J&J know that a lot of what we're doing is partnerships with the academic community. And whether it's biomarkers or screening studies or development of, doing some early stage research, right? So again, I think there's a lot of strengths within the academic community that we, in the industry don't have the resources to do. And so I think, again, just this idea of open collaboration, understanding, what that pathway looks like from an industry perspective. And maybe you want to have Matthias and others back for a seminar on exactly that, because we could probably fill the whole hour with some of the, where industry, what we think about and how we think about things maybe a little bit differently in a complementary fashion, right?
Yeah, no,
to
that's a
the
great
academic
idea.
science.
That's a great idea. And actually, it would be nice to see where are the gaps on both sides and how that they can be overcome together. I think that's a really wonderful idea. Because we're all, in this space, I think everyone is united in trying to like figure it out, figure out the etiology and figure out the remedy. Would anyone else like to offer, some kind of anything more about, endotypes or phenotypes or genetics, or putting the
Can I
piece.
possibly just add something onto kind of Jessica's kind of request for kind of industry and scientists to work together? I think one of the kind of useful areas that we could focus on is looking really at the mechanisms of actions of the different immunotherapies that you think are going to be potentially promising in type 1 diabetes, and really focus on identifying the key targets of those. And then within our endotypes that we're seeing, for example, within the pancreas, looking at what's happening to those key targets in the different endotypes to try and identify whether particular therapies may work better in a specific subset of individuals.
That's well said.
I want to maybe bring up another thing as we think about different patient populations that we haven't touched on, and it's something we think about a lot at J&J, and this is the idea of diverse populations, right? So you look at the latest search data that shows that the largest relative rate of increase of type 1 diabetes is in African Americans, and as we think about specifically, let's talk about screening for type 1 diabetes, and the genetics has been largely based on populations from Northern European descent. But as we think about applying something like a genetic risk score to the broad US population as an example, what does that mean? And then you start even driving down to pathogenic mechanisms. I don't think we have any understanding about how race and ethnicity matter. So maybe also just a call for action to the academic community. I know some of you are already thinking about this, but I see this as a major gap when you think. You asked the question, Monica, about where industry is thinking. And if you look at the timelines for 10 to 15 years for a drug to make it to market, if we're looking 10 to 15 years out, this is a major gap, I think, for all of us.
Yeah, that's.
And I don't know that we have easy answers to how to get there.
It's an excellent point. And I'm thinking you need to have more tissue repositories inclusion also. I just wanted to bring up a couple of questions that are still here in our chat. And one is, I just sort of brilliant talks. Are those TIDE 1 and TIDE 2, are they genetically different? I think Sarah's typing an answer. Here's another question. Is anyone planning to modify their research based on studies showing more type 1 diabetes diagnosis after being infected with COVID? Anybody out there in the panel?
I would just say, I'm not sure that there's like clear epidemiologic data that there is increased type 1 diabetes after COVID. That data that the CDC recently published was not broken down by type 1 and type 2 diabetes. And I think, you know, based on what they presented, it's hard to kind of tease out how much of that is related to changes in presentation and you know, lifestyle changes that might have been happening in children who are at risk for type 2 diabetes. So definitely, I know Annetta Ziegler has published some data about autoantibodies in the pandemic, and incidence of that has not really changed. So I have not personally seen convincing evidence that type 1 diabetes has been increased by the COVID pandemic. That's my thoughts on that.
Okay. And then I'm going to just grab this other question. Thanks for the question from Teresa Rodriguez Calvo. The problem I see that is that most new therapies are tested first in adults, where they might not be as effective as in children. If age really makes a difference, how can we convince FDA and regulatory agencies to go beyond adults? Is it really feasible? How can we link age at diagnosis to clinical trial to therapies? It's a big question.
Really hard, Teresa. They want to see safety data in grown-ups first for these therapies that are, you know, you're imparting some risk in people who you don't know for sure are going to get disease, right? So I think it's tough. I agree with you though. It's so important to look in that population.
Yeah.
I think a similar challenge that we have is that we are often thinking about in clinical trial design, testing a new mechanism at stage 3 onset, when we clearly sort of know that probably people are primed to respond earlier in the disease process. So that is just because it's a pragmatic approach that we've taken. It's easy to identify people at stage 3 onset. And then the only other point I would kind of make about, you know, how we might use some of this knowledge that has been so beautifully articulated here today to better design trials is this point that made by Teresa Quatron in the chat is that, and I think we're incredibly fortunate to have people like Jess and Matthias who are partners and care deeply about type 1 diabetes as a space. But there's a lot of companies that are very hesitant to even come into the market because of very compelling business cases, et cetera. So, and because a lot of things that we've tested have just not worked for a variety of reasons, right? So I think that part of that response of these 30 years of this uphill battle has been, you know, articulated here in this idea that, well, maybe one of the reasons things don't work is that we're not quite hitting that heterogeneity. So I think that it's been a response to that. Clearly, we can't, you know, we've got to move forward, I think, in both parallel streams, trying to understand the heterogeneity, but then also just really trying to get a treatment.
Yeah, that's a perfect summary, really. And I mean, I guess I would also offer that, you know, at first breast cancer just seemed like one thing, then it became, you know, many different stages, and maybe there's some things from that model and how it progressed that we can, or that, you know, the field can incorporate as it tries, although, you know, again, it's a market-driven business, so it remains a small market. But that's not to say that, you know, discoveries in type 1 diabetes in the laboratory couldn't be parlayed into other disease states, other autoimmune diseases, and I mean other diseases in general. So there's a lot of room for discoveries to come from exploration in the laboratory on type 1 diabetes. I guess.
So Monica, if perhaps I could just come in. So I completely agree with what Carmella just said there. And I think, you know, it definitely has been an uphill battle, but hopefully we're now at a point where we're primed to be in a better position to get more information from the trials that are coming online now. And I just wanted to make the point that I think we have to consider all of these different parameters together. So when we were talking earlier, Monica, about sort of whether the genetics was wedded to the biology and whether, you know, phenotypes reflected the genetics and so on, I think it comes back to this point about type 1 diabetes being a polygenic and complex disorder. So the genetics is going to give you the susceptibility, but that susceptibility is not necessarily going to translate into disease. And if we just think about our very simple CTLA-4 haploinsufficiency situation, we have first-degree relatives that have the same mutation and no disease. So, you certainly can have this uncoupling between susceptibility and whether you get, whether it's the environmental trigger or the other genes or, whatever it might be that brings you into the sphere of having the disease itself. So I think we have to take all of these parameters, study the biomarkers, the immune phenotypes, the genetics, looking certainly at age of presentation. I think for me, that's the strongest indication we have of an endotype so far. And wouldn't it be amazing if that turns out to be a proxy for, a mechanistic difference? So I think we have to take all of these things together when we're looking at how we interpret this and, ultimately decide how to treat different subgroups.
Well said. And we are at the top of the hour now. I think, we have 5 other sessions ahead, 5 other events in the State of the Science series, culminating in the very last one, which is sort of talking about the roadmap forward and how to bring all these data sets together, which is sort of a nod to what you just said. And that will be March 10th, but we hope you can, we thank you all for attending and for driving discussion. And we look forward to our next interactive towns hall style State of the Science series that will be happening on January 27th, where we're going to discuss cutting edge technologies and in silico solutions for the type 1 diabetes laboratory. Thank you all again very much, and hope to see you again in January 27th. Have a great rest of the day.